Hello, everyone. Thanks for coming to this week's video lecture. Today, we're going to start on Unit 3 and specifically on Chapter 13, where we're going to cover microbes on the human body, where microbes typically live and how they can be transmitted into potentially problematic regions and how they can establish infections. And with that, we can get started. And so the human body has regions that are not sterile, that are colonized by tons of different kinds of bacteria, fungus, and protozoas and other microbes. And so um, we can characterize all of the microbes that exist on the human body as the microbiota. Or the microbiome. And this is all of them that exist on the human body. And most of the time, these bacteria are commensal, where they do not cause harm to the person that they live on. This is not always the case when things are dysregulated, where maybe a bacteria might get into like a part of a wound and create infection, and they're kind of opportunistic. And so we'll kind of learn that there are opportunistic pathogens that can establish affection when a host is weak or when the opportunity arises. But most of the time, the microbiome is made up of non-pathogenic microbes that colonize the skin or other tissues that and they just hang out. They just go about their normal lives, and they don't really cause any harm to the host. And in this case, it's called commensalism, and there's a commensal behavior. There's also mutualism, where both organisms in the relationship benefit from one another. An example of this would be like in the gut, where you would have, for instance, a gut bacteria that you eat food and that nutrients goes in there, and you're feeding that bacteria that nutrients. So it's benefiting from you. And then it takes those nutrients and breaks it down into vitamins, hormones, and other compounds that we would be able to use. And it's a lot easier for us to use when they're already converted in that way. And so then we benefit from them. And so there's the mutualism, where both of these parties are benefiting. But not all of these tissues in our body are not sterile. Some of them are sterile. For instance, our blood, our liver, our muscles, our bones, these places, there aren't bacteria. And when bacteria get into them and establish a population, they start to cause tissue damage and create an infection and cause disease. Now, something that's really interesting is that the places in our body are each unique and different. There are different conditions. For instance, the pH might be different, the moisture content might be different, the oxygen environment may be different. And this, just like how the environment affects like which plants grow around. For some plants, they can't grow where it's cold, and where some plants they can't grow where it's hot, or there's not enough water or something like that. The same thing goes with these microbes. Is some microbes like certain conditions and other rather than others. And so when you sample across our body, these microbiomes within specific locations are actually have different profiles or different sets of bacteria that live in those regions. There might be some overlap. However, the profiles are fairly distinct. And so that's a kind of a neat thing. We also have this kind of benefit that these bacteria are not pathogenic, but they colonize our skin. And so they're taking up space and residency. They take up residence and take up space within for instance, on your skin. And it takes up the resources in that area, so that if you did get a pathogen there, it would be a little bit harder for them to move in and establish an infection and start using up those resources. And so this is an idea that's called microbial antagonism, is that our colonies of beneficial bacteria are helping in such a way that are helping to like deter potentially problematic microbes from taking hold and potentially establishing infection. And this is really seen or like talked about a lot of the time within the gut. And so we talked about how different locations on your body have different profiles of microbiomes. They have different microbiomes. And this also takes, and part of this is because of the types of environments that are being made. And so for your skin, you have the top of the skin where you have the epidermis, you have the you have the epidermis, and you have the dermis and the subcutaneous layer, where the epidermis has like really keratinized cells, really tightly packed together that go up in multiple layers. And within this layer, you have the top layer, which would be the least sterile, and you start having bacteria within this region. But once you go down into the tissue, it becomes more and more sterile because of those extra barriers that exist within those keratinized epithelial cells. But there are follicles and pores and ducts that exist on your skin, like hair follicles or oil glands. And oh, this one's a sweat gland, but there are also oil glands that exist that are also colonized by bacteria. And when certain bacteria get in there, or when that microbiome is dysregulated, that's when you start getting those infections. That's when you start getting those irritated marks on your skin. Is because the microbiomes within these regions are dysregulated, or something has taken hold that isn't supposed to be there, and our body has identified that it's not supposed to be there and is responding to that. And we'll talk about those responses later. But just to show you that this is kind of what the colonization looks like of the skin. And then for the respiratory tract, oh, this isn't the respiratory tract, but kind of the respiratory tract is here. Respiratory tract, you have the nasal passages and nasal cavities. You have the what's it called? It's even labeled, why don't I just call it the esophagus? The naropharynx and into the larynx and the bronchioles. But eventually, there is cilia that are down in the bronchioles and up through the trachea. And as you go through these regions, these cilias are actually, we're going to talk about how these are physical barriers that help prevent microbes from getting in these regions. And so as microbes get in, the cilia is going to be forcing and help clearing these microbes. And so it kind of starts to sterilize that tissue as we go into our lungs. And that's how we can keep our lungs sterilized. We also have, we'll talk about how we have like these, we have mucus and saliva that also helps to trap and eliminate those microbes in this region so that we can control them so that they don't get out of control and create an infection. Back to the gastrointestinal tract. So you have your mouth, to consume, you have a completely different like environment in your mouth. You have saliva going on, you're constantly intaking nutrients, or if you're drinking like sugars and things like that, you're constantly feeding nutrients into your mouth. And so there's lots of bacteria and things that are happening there. But that's a very different environment than, for instance, in your nose, where you're not shoving sugar into your nose, hopefully. But then that bacteria, goes down into your esophagus and then into your stomach, and then your stomach has stomach acid that has an incredibly low pH that will mostly destroy most bacteria that end up in this. And this is how we can start to sterilize our food intake and sterilize the contents and keep our small intestines sterile. And so it's really interesting that we have this really long tube of where we have nutrients come in, and then we have excrement come out. And really the only parts that are colonized with bacteria are the very beginning parts before the stomach, and then the very end parts with the large intestines and the colon. But the middle part, from the stomach all the way to the small intestines, is sterile because of that stomach acid and the conditions that are being maintained. And so when bacteria can make it through the stomach acid, it can take hold in this region and cause infection because it's not supposed to be there. And so to prevent that, there would be there would be defenses most likely that would help to clear those kinds of infections in those regions. And we'll talk about those later. I'm not sure if it's this lecture or the next lecture, but it's in this unit at least. And so each of these, we talked about how each of these locations kind of have a unique profile of the microbiome. They have different bacteria that live there. And so in the mouth, you have things like yeast, you have streptococcus and lactobacillus, you have Bacteroides and oh, and this amoeba. This is a really interesting one. This is an amoeba that lives in the like crooks of your gums. But these are specifically made or like specifically have evolved to live in your mouth, and they thrive in that environment. But if you put them in a different environment, if you try to put them in your ear, they wouldn't have as good of a time. And so it's just really interesting to kind of keep in mind that these locations have these different profiles, and what's causing those different profiles is the environment that they're in, which is like directly resultant of like the physiology of that tissue, right? Like we are constantly excreting saliva, which has enzymes that can help break down proteins. So these bacteria have to be able to withstand that. They have to withstand the like mechanicalness of like us cleaning our teeth, of us like just like having things in our mouth constantly. They have to be able to withstand the or like take in the wide variety of chemicals that we have, and be able to survive in an oxygen rich environment because our mouth is highly open to oxygen. But for instance, in our gut, you don't have oxygen as much. And so the bacteria that's in your gut needs an oxygen poor environment and won't do very well in the mouth because it is an oxygen rich environment. Here we go. Large intestines, you have Bacteroides, you have bifidobacteriums, Clostridium, Staphylococcus, again, some yeast, you have E. coli, and like Escherichia coli, Enterobacter, Citrobacter. Those ones are important in creating metabolites for us to take up, and those would be used for like hormones, metabolism, or for signaling or amino acid or vitamin production. Sorry, I'm very or right now. We talked about the respiratory tract already. For the urinary tract, you have you have the vulva that has bacteria all the way through it. The urethra's opening has bacteria in it, and then eventually it becomes sterile at the very end. So when you're putting in a catheter, you have this risk of taking this bacteria into the further into the urethra and causing a urinary tract infection. And then you have the vaginal canal, which is really interesting because it's an aerobic environment. There's no oxygen in that environment. And specifically, the bacteria that live in that environment thrive in an oxygen poor environment. And as a result of that oxygen poor environment, they do fermentation and create acid. think it's lactic acid, but they create acid, which lowers the pH, which creates a acidic environment within the vaginal canal, which is helps deter other bacteria from taking hold in that region. And it specializes that so that, like, for instance, the bacteria outside on the vulva isn't able to grow inside of the vaginal canal, and it helps prevent those infections. On the testes, you have the bacteria all the way around the outside, as well as, again, with the urethra. And again, with when you catheterize, you can take that bacteria into, or yeast into, or like fungus into that urethra and create an infection. And so you have to be careful with things like that for people who do catheterization for a long time, because it is it drastically increases the risk of those infections because of how these microbiomes are set up. And so, excuse me, I need to. Sorry about that. So now we're going to talk about how these bacteria are leave a place where they're supposed to be into a place where they're not supposed to be, and create that are potentially problematic and create potential damage and infection and disease. And so this part has a lot of like terms. And so you have just a pathogen, which is a microbe that can cause infection, and its pathogenicity is how severe or how capable that pathogen is at creating those infections and establishing and making that disease. Because some microbes are very good at it and highly specialized at it, and some of them are not very good at it. There are true pathogens these are those pathogens that are very good at it, that they have very high pathogenicity. These are things like the bubonic plague, like malaria. But then you also have opportunistic pathogens. And these are those pathogens that we kind of talked about that do sometimes exist on the skin, but they don't cause infection unless they are given that opportunity and they take advantage of it. And so maybe the person has a weakened immune system. Maybe there's a gash in their skin, and they're like, it's a chronic wound. And so there's some issues that make it where it's like has a weaker immune like response to it. Um or another one is if you just consumed an actual ton of the pathogen, it just has given, it's just like you've consumed enough, you've taken a large enough dose, and we'll talk about that a little bit, to initiate infection, even though that typically you would not be exposed to enough to cause infection. An example of this is Serratia marcescens. Serratia marcescens is typically not pathogenic. However, if you do have a very weakened immune system, or if you have a large quantity of it, if you inhale a large quantity of it or drink a large quantity of it, it could potentially take hold in specific tissues and cause disease. I had a lab, one of my friends at one point, they were using a laminar flow hood that had a liquid culture of Serratia marcescens that happened to have spilled into the filter. And so typically you wouldn't have infections from that, from just like being exposed to Serratia marcescens at just a reasonable level. However, because it was in the laminar flow hood filter, it was constantly blowing that bacteria into their faces, like out of the hood where they would be sitting, because laminar flow hoods are not specifically designed to protect the person. It's specifically made to keep the field of sterility within the hood. And so it exposed them to so much of that bacteria that it actually did cause them respiratory infections from that. And so that's an example of that opportunistic infection. You also see opportunistic infections in hospitals a lot, where someone will come in for a specific infection, they'll be treated, but in that process of being treated, they will come up with a secondary infection that is often, or doesn't have to be often, but like can be this other bacteria that you typically wouldn't see cause an infection by itself. And so those are typically called opportunistic. And so anyway, you also have different factors that affect how well the bacteria or microbe can cause disease. You also have how well the host can defend from that disease. An example of this is age, your genetic profiles, and other things like confounding health issues. And so you have that pathogenicity that we talked about. How capable is it of creating harm and creating that infection? And then you have virulence, which is how is kind of similar, but it's how readily can a pathogen infect its host cell. And virulence factors are essentially those confounding variables that causes the bacteria to have better advantages comparatively to the general bacteria or general pathogen. Because you can have, for instance, like an E. coli that can't cause bacteria, that can't cause infection, and then you can have E. coli that can cause infection and then can be pathogenic. And it's because they have different virulence factors that up their virulence and make them more pathogenic. And so examples of this are potentially specific enzymes that help degrade something that allows them to like get into the body easier, or maybe it's something like it has different sugars on the outside of its cell wall, and that allows it to evade the immune system for some capacity. And so for these bacteria to get into a tissue that it's not supposed to and cause harm and establish a population and create an infection, first it has to get to that location. And so we're going to kind of skip the part of how it's transmitted to the person, but eventually it needs to get into the individual. And this is called the portal of entry. And this is typically what holes do you have? It can be your nose, your eyes, shoulders. Wait, that's not all your holes. You have your eyes, ears, nose, and mouth. Yeah. And then you have your anus, you have your urethra, and you have your vaginal canal. And then you would have any like extra holes that you provide, right? Like if you had a subdermal piercing, that would be something. Or if you just had like a regular piercing, that's a hole. That would be a hole into your body. If you had just like a wound, a scrape, all of those things are breaking the barriers that your body has and allowing for a potential bacteria to get into that tissue and potentially cause infection. And so these are called the portals of entry. And so some bacteria have very specialized portals of entry. They don't just like come in willy-nilly. Some are specifically geared towards the respiratory tract. And so they come in by the respiratory tract. So let me think. So I think, so an example of this would be SARS-CoV-2 is a respiratory tract virus. It is it enters into the respiratory tract through through that it's transmitted. It gets to us. We'll talk about how it gets to us in a minute. But then it goes into our portal of entry is our nose, goes into our mouth as we breathe in. Then it goes into our respiratory tract. If it gets past all of those barriers that we have, and it causes infection. An example of something that has more generalized portal of entry is like anthrax, because anthrax you could inhale it and you can get in a respiratory anthrax. You can ingest it and then you can get gastral. I think you can get a gastric form of anthrax, I believe. Um, and then you also can get an infection of anthrax on the skin. And so that pathogen has multiple forms of disease, and it's because it's colonizing different tissues, while other pathogens can only target certain tissues. And that's partially because just like how our oral microbiome can only survive in our mouth or survives best in our mouth, these pathogens also have their best and preferred like tissues for infection, which makes it nice to study like how disease works because it is more reliable and it's not just like a random and willy-nilly. But anyway, so the bacteria need to get into our body, and then they have to get past all of our barriers and like for instance, in our respiratory tract, we have cilia that's constantly moving and trying to get all of this debris and bacteria out of the respiratory tract. And so if you're trying to invade and you're trying to infect the respiratory tract and all the cilia is moving, you're going to have to hold on and try to avoid being scraped out or pushed out of the tissue. And so microbes have different organelles like fimbriae or different things like glycocalyxes or different structures that help attach the microbes to a surface. And those might be like physical attachments that are like more like the fimbriae, which are like hair-like structures, or in the capsule, which is kind of like a sticky, kind of like sugary thing. Glycocalyx, it's a sugar protein, like sugar protein mixture. Or you can have things that are like protein-protein interactions where they're actually like holding on through like bonds, like molecular bonds between two proteins of the bacteria and like the host. And then once you have like actually attached into the tissue, you have to then start dividing and replicating and living your life. And eventually the host is going to notice you around the time that you're causing harm to the body and harm to the tissue because you're growing so well. And then eventually, hopefully, you're able to clear that infection and that infection is resolved. Sometimes those infections aren't resolved, and that goes from like an acute infection to like a chronic infection, which usually has different like symptoms and like pathology profiles related to them. And so we talked about the different portals of entry, right? We have our eyes, we have our ears, we have our nose, mouth, we have any like piercings that we would have. You have the vagina, the urethra, you have for the baby, I don't remember that one. You have the bacteria that's in the vaginal canal, but then you have the barrier of, I believe, the cervix that keep this protection. But then you also have the placenta, and the placenta has barriers that help prevent the bacteria to get into and infecting the baby. But for an infection into a baby, they would have to at least get through that portal of entry there or through the blood. So anyway, when you are studying these diseases, you realize that in theory, one bacteria could go in and create this infection. But there are all of these things that are barriers that are trying to stop that from happening. And so if you just have one bacteria that come in, one of those barriers may stop that bacteria. And so so you can kind of quantify how many bacteria you would actually need to go through this system, this path of going through these barriers and making it to like the site of infection, the site that you want to like attach to, and then starting to grow and establishing infection, you know, how many bacteria do you need to provide to actually get that infection started? And this is like quantified as an infectious dose. And this is dependent on each bacteria. Each pathogen itself has its own infectious dose, and it depends on the things like its virulence factors and its pathogenicity, and the host immune system, on how readily a bacteria is able to go and how many bacteria you need to get infected. And so we talked a little bit about how once you've got that portal of entry and how many bacteria you need to get in, that bacteria needs to attach and start to colonize your tissues, and it can do that through different structures, and it can do that through different like molecular interactions as well. We talked about how virulence factors are increasing like the probability of that bacteria to like cause disease and establish infection. And other examples that I didn't talk about is potential toxins that are problematic to the host, that are damaging to the host tissue, but that remodel the location of infection to make it where it's better suited for the bacteria itself. And the other thing is like the capsule, that glycocalyx, it helps avoid detection from the immune system, but it also has this helpful thing that it helps kind of prevent the phagocytes to phagocytize or like swallow the bacteria. And the way it does this is by avoiding detection. Still the same thing, but instead of like avoiding like detection from like an antibody or something, it's avoiding detection from like these phagocytes or like macrophages or something. And so these toxins that these cells can produce can cause multiple different things, and it usually specialized to the disease and the pathology that the tissue that the pathogen is like specialized in. So for example, ringworm, which typically happens on the skin, and it creates like a really a ringed dry spot that's flaky. Part of its one of the toxins that it produces is keratinase, which is an enzyme that dissolves the keratin. That is, if you remember when we talked about the epithelium, epidermis, sorry, the epidermis of the skin, it has these epithelial cells that are keratinized, that have a ton of keratin in them, that are being held together in this like tight little like stack. And these layers are helping to protect our body from infection. And so if you're able to dissolve the keratin that's holding these cells together, well, first off, they're going to slough off, which is where you get that dry sloughing off from. But you can also penetrate into that tissue deeper, because you're essentially removing layers and you're kind of digging down deeper. And so if you had a ringworm that didn't have that keratinase and didn't have that toxin, it would have a lower virulence. It wouldn't be able to create as severe of an infection as a ringworm that has that keratinase. And so that's an example of how like a virulence factor would come into play. But you have all kinds of different enzymes that are specialized at defeating a host immune system or avoiding a host immune system. Another thing is like the ability of creating like biofilms and like the the community that can be formed like in biofilms on like the teeth and stuff. Having specific genes that are beneficial to that can help also bolster like the probability of like a pathogen or a problematic bacteria like surviving in a region. Those biofilms don't happen just on the teeth, they can happen in other tissues as well. So when we talk about toxins, you can have toxins or enzymes that are that are secreted into the environment that are specifically to do work to remodel the location or the environment, whether that be signaling molecules to be, oh, that's not what we're doing. We're not signaling molecules. It is excreting signaling molecules, and that might be a defense that the bacteria is using. But these enzymes are doing work. They're acting on something. For instance, the keratinase is a enzyme that is being excreted into the environment to dissolve these joints between the cells or the keratin between these cells, and allowing them to be able to infiltrate deeper into from the epidermis into the dermis layers of the skin. And so you have these enzymes that are excreted out of the bacteria and can do that. But you also have endotoxin, which is held inside the cell wall and membrane of the bacteria up until the bacteria is killed. And when the bacteria is killed, that cell wall is destroyed and degraded, and it releases this enzyme called LPS, or a lipopolysaccharide. And this LPS causes a inflammatory response in the innate immune system of a host, and it can cause toxic shock syndrome at large levels and cause death if it's provided too much, but it stimulates the immune system. And so the LPS does its own job. It's not like it's like this hidden bomb that's been designed to kill us because we killed it. It's not like a revenge tactic or anything like that, but our body has developed a response to LPS because we know if there's LPS, bacteria has been there, which means there's bacteria probably still there, and we need to mount a response to it. Or there's bacterial debris and we need to clean it up. And so we need to mount a response to it. And so there's a host response to this LPS, which makes it a toxic molecule. And so that toxin is held specifically in the within the cell wall and the membrane. And just like the glycocalyx and the sugar proteins and things like that, this is also a lipopolysaccharide. So this is a lipid and a sugar complex. So it's a lipid and branched sugars. But it is held in the membrane of those cells. So here's an example of that bacteria excreting out these toxins and then getting into the cells, or them doing some work to remodel some tissue, whether that is to help them get in to create an infection, whether that be to like kill the immune system or whatever. You also have toxins that have specific mechanisms, like this one here, which is called an AB toxin, where you have this binding subunit and then you have this active subunit. And so different bacteria will have different AB toxins, but just the general gist of like there's this subunit and this subunit that is shared across multiple pathogens. And it's called an AB toxin. And so you have this binding subunit that can bind to specific cells. And because of molecular interactions, it can only fit specific cell receptors, and not all cells have those receptors. And if the cell doesn't have those receptors, the toxin can't bind to it. So now we have a toxin that's specialized to a specific cell. This binding subunit binds to it, it creates an endocytosis, and it engulfs the toxin into the cell. In this, it creates, so an example is maybe this is an endosome, so it starts to become acidic because it's going to degrade this, and it breaks apart the AB subunits. And now we just have A floating around and we have B bound. Then A is able to export out of this vacuole, or for instance, endosome, and actually do like a signaling cascade or do some kind of like trigger some kind of transcription response to this activation protein is present. And so it can cause, so in this case, it's actually attacking a part of the, what is that? That's, so NAD is a high energy carrier, and we're creating ADP. I think this is a part of the Krebs cycle. I don't remember, but it is impeding, nonetheless, it's impeding some cellular system. And so that impeding of the cellular system either kills the cell or it changes the cell's morphology to make it more like beneficial for that bacteria or pathogen survivability. Sometimes this could be turning on or turning off specific transcription. What is the words? Transcription gene sets, essentially, like downplaying viral, like your antiviral response. You can like downplay that by turning off certain regulatory systems using certain toxins. I'm getting into the weeds, so I'm just going to keep moving on. Okay, so kind of already talked about how these toxins are kind of doing work and then how our body is responding to these toxins. Sometimes it can create an immune response, like the LPS creates an innate immune response, or you have pyrogens that can stimulate a fever. And so when you start having an infection, clinically, you can characterize these infections and the pathologies in a few different ways. You can talk about the patient itself, and the patient's experiences are called symptoms. So that would just be like, I'm feeling this way. Then you have what the medical provider can observe just visually and objectively from the patient. And these are signs. This would be things like blood pressure. This would be things like iron level in the blood. This would be things like fever. This would be things like they look pale as fuck, you know? These would be signs. How is the patient feeling? Those are symptoms. And constellations of symptoms and signs are designated as syndromes or different like disease profiles or different pathologies. And so you can use the different symptoms and signs that different patients have to identify and characterize these pathogens and try to identify what disease they have. And so you can have infections that are localized to a specific location within the body. You can have systemic infections that are throughout the entirety of the body. And then you can have focal infections, which start in one location and go into other locations later on. An example of this with like cancer would be like malignancy, but with pathogens, it's called a focal infection. And so you can also have, just like on a plate, you can have a mixed culture or you can have a pure culture of infection. If your appendix bursts and it infects your abdominal cavity with bacteria, you're going to get sepsis. And there's going to be a ton of different bacteria that exist in your appendix when it bursts. And it's going to inoculate a ton of different bacteria into your abdominal cavity. And so those are going to cause infection. And because there are multiple different species, these are called a mixed infection or a polymicrobial infection. You also have like a pure culture infection, right? Would be the opposite of that, where you just have one specific culture of bacteria that is causing a problem in a particular region. If it's a particular region, it's a localized infection, right? We talked a couple of times about how you could have an infection and then eventually you might end up with a secondary infection with a opportunistic pathogen because you have a weakened immune system and a weakened response. And so this is the terminology you would use. That first infection is called a primary infection. It's that first infection that's causing that weakening. And a secondary infection is a second time that comes back and creates those infections. If it is a different infection each time, those would just be like reoccurring secondary infections. But if it's the same infection that's lasting for a long time, it would be considered chronic. Most times infection happens and then it's resolved, it's acute, and it happens once and then it's done. But then you have chronic that lasts a long period of time or it repeats over and over again. So when a bacteria gets to you, it goes into your portal of entry, it gets to your into your tissue, and it enters this period of incubation. And this is the time where the bacteria is attaching, it's kind of getting comfortable, it's getting settled in, turning on all of its genes and responding to the environment that all it needs to do, and starts to divide. And eventually, this incubation phase goes into the sorry, prodromal stage, where the bacteria is starting to rapidly divide and starting to create some slight problems. This is where tissue is starting to become damaged, and the individual and the host is starting to feel that this infection is starting to take place. Your body is starting to respond at this point, and then you get this period of invasion where the bacteria and is ramping up. It's the body's response is wrapping up, and well, the body's response hasn't really like caught up yet. It's the body's response is still kind of working itself out. It's kind of like someone may have been notified that the that the infection has happened, but maybe only like 2 or 3 people know about it right now, you know. But anyway, the pathogen is like really wreaking havoc and is starting to cause real damage to the tissues. The patient is truly understanding that they're sick and they're actually um, experiencing a lot of those symptoms that you would see, I guess, that they would describe, and you would see the signs technically. But eventually, your immune system is going to catch up and is going to start trying to get a handle and curb this infection. If your immune system just never responded, this period of invasion would have just skyrocketed until your death. But because our immune system is taking hold, it curbs this infection, and this is the height of the infection, and it reaches into a period eventually of remission, what is called the convalescent period. And this convalescent period is where the immune system is starting to eliminate these bacteria. They're starting to destroy them and clean it up. And then towards the end of this, you're going to start to remodel those tissues and repair the damage that was done to those pathogens and keep clearing the debris that was left over. And so when you're responding to these infections, there's a couple of different cells that work to, sorry, it's a little bit later. Where is that?
Gotcha. Okay. That's a little bit later, apparently. So you can characterize these infections. Sorry about that. You can characterize these infections in a couple of different ways. It depends on, um, sorry, that really threw me. Give me a second. So you learn about what these cells are that are in the immune response in the next chapter. And so for this chapter, we're just trying to describe these infections and how we can clinically describe their pathologies. And so we can describe these infections in a few different ways on where they're actually occurring and what tissue are they occurring in, and also um, um, what kind of like measurable effects that we can, for instance, characterize. And so, for instance, you have leukosis, where we have an increase of white blood cells, or leukopenia, where we have a decrease of white blood cells. And we have, depending on where those bacteria are occurring, you have different names that determine where that growth is happening and what type of organism is doing that growth. And so you would then also be able to have things like you'd also have things that would be like physiological symptoms, physiological signs, like for instance, iron level in the blood. And you'd have the terms that can describe those independently. You would have then also, it's interesting that sometimes these host immune systems are different from person to person because their genetics are different, and they have different immunological histories. And so sometimes you can have people that have infections, but they don't actually show the same symptoms in the clinic or in the world, because there are some times where you have people who don't show signs of this infection. We saw this with carriers of SARS-CoV-2, where there were people who would get sick, but they could just live their normal life. They might feel a little bad, but they can just go about their life asymptomatic. And this is potentially problematic, because when you don't have the symptoms and you're not feeling bad and you're not taking it easy and not being mindful of that you're potentially infectious or contagious, you expose those around you to potential pathogens. And so in this process of getting into the body and starting the infection, and so right here you have this period of incubation. This period of incubation is typically described as the latency of how long of the period of incubation describes specifically like the bacterial growth. Latency is kind of the patient's experience, but they're capturing similar things. And it's essentially like how soon after that like bacteria has been exposed into the person, does those symptoms convalesce? No, when do those symptoms appear? And so this is called latency. We've also talked about how some infections are chronic, how they can just stay and never get resolved, or we can see how there's like maybe a secondary infection that's just like a reoccurring infection. And these are called chronic carriers, where they have an infection, but they constantly have an infection, or they have infection commonly, and they are consistently contagious, which is potentially problematic from like a public health standpoint, right? Like things would have to be like, obviously we can't like say you just can't go out in public, but things and efforts need to be made on understanding these kinds of conditions and how to manage them from a personal health standpoint, but also from a public health standpoint. And so we kind of have talked about like how these bacteria just kind of get into the body and then how they take hold into a tissue and cause infection. But now we're going to talk about kind of how they get to those points. So just like how we have microbiomes that are specialized to different locations in our body, you also have the same kind of thing within the world, just like with animals and plants. And different insects, you have specific locations that are specialized to specific species. And the same goes true for pathogens. Some pathogens are specialized for water, and some pathogens are specialized in soil. Some are dependent on the host itself and can only live on that organism. And so those locations, those native regions, um, native growth ranges, is that what it's called with plants? Yeah, they're native ranges. But like that natural location that these pathogens live are called their reservoir. This is where you would just like typically find them on your day to day. And somehow these pathogens go from their normal place or these reservoirs and are transmitted onto humans, into the port of entry, onto these tissues, and create infection. And this is done through vectors, where you have something that can like quite literally carry it. For instance, like with mosquitoes, where they can carry that malaria. Or you have fleas and that could have like your Yersinia pestis, for instance. And those things that go from this natural reservoir of the bacteria onto a host. And those are called vectors. And these vectors can be alive or they can be mechanical. Those mechanical vectors can be things like um, let me check if I I think malaria just I think mosquitoes are mechanical vectors for malaria. I don't think the malaria actually I don't think malaria is the paramecium needs I don't think it has a life stage and mosquitoes can. I was wrong. So the mosquito does have a role in the malaria's life cycle. So malaria is a plasmodium that gets into the mosquito and it grows into this stage before it goes into humans. And so the mosquito is a biological vector of malaria because it is important in the life stages of this plant. An example of like a mechanical vector that where like the thing that's transmitting it, the vector, isn't necessary for the life stages would be, for instance, tobacco mosaic virus, for instance, or other mosaic viruses that are passed from plant to plant. Sometimes you can have people are handling the crop and they'll get it on their hands and then they'll go to another crop and they'll look at it and they will actually transmit and infect another crop with that mosaic virus. And so that is a mechanical vector that we don't participate in that virus's life cycle comparatively to malaria, which like goes into the mosquito and then does some living stuff, which is the biological part. It has a part of its life cycle in it before it actually causes that infection. Yeah, so I skipped over this part where when you have bacteria where were we when we started about this? And so we kind of talked about how earlier with this chronic carrier, a person that has this like chronic infection is like constantly contagious, essentially. They're called a carrier, where they're able to have this pathogen and they are able to pass it to someone else. And sometimes those carriers are sick and they're just sick and they just pass it to someone else. This is, just random having kids around. This is very common. Asymptomatic carriers are people who may have an infection, but they don't show that they have the symptoms of the disease. And they might not feel very bad, or they might not show that they feel very bad. They might carry along just fine, which is particularly dangerous because, again, if you don't know that they're sick, you can't make those precautions of what you do when people are sick. And so it presents a potential risk of getting infected. But those are called asymptomatic carriers. And then you have passive carriers, which are able to transmit a pathogen from one to another without actually interfering. Again, this is like that mechanical vector without participating within the life cycle of the virus itself. So there are specific pathogens that are designated to specific species. They're like, we infect humans or we infect bats, and that's all we do. And that's great. But there's also specific pathogens that are generalized pathogens that are able to not only infect one specific species, but infect multiple different species. And this can sometimes be problematic when it comes to wildlife or just any animal and humans, because there can be a transmission of disease from those animals to the human, and this is called zoonosis. And this happens fairly regularly with things like rabies or things like leprosy, or or like ringworm is another one that happens a lot. If you snuggle with your cat or something, you might get ringworm, or if you go, I guess, running around in the dirt also. But we're talking about zoonosis. So if you're cuddling with some animals, you might get ringworm. And it's because it's passing from the animal to the human, and this is called zoonosis. Here are some examples of that, and this is the typical reservoirs. This is where you would typically find them, and this is how they get around and are transmitted to the us or to the host. You have some of these take specific vectors like ticks or mosquitoes, and other times it's just the environment and like aerosols and things that can help disseminate these pathogens around. So we have the soil and water as really common places for bacteria to live and grow. A lot of our pathogens that we talk about are in the soil: Clostridium botulinum, tetanus, anthrax, round this hookworm, but also like ringworm. Also, it's a big one. And so you also have diseases and infections that are able to pass from human to human or from individual to individual. And then you also have infections that are able to be, you went into a cave, there was fungus about in the air, you inhaled that, and it got into your lungs and you got infected, but you're not going to breathe out fungus spores and get someone else infected. That would be non-communicable comparatively to SARS-CoV-2, where you catch COVID, you get infection into the lungs, you start producing a ton of virus, you start shedding that virus in droplets and in aerosol vapors, aerosol droplets, and then someone else can catch it. And so that's communicable. It's contagious. And this can go from direct transmission, just like with SARS-CoV-2 with aerosol transmission, like coughing and sneezing, or just breathing. And also, you can also have indirect transmission. And so like this would be something like coughing in your hand and like touching a surface. And the surface is called a fomite, or a vehicle that helps facilitate that transfer. And so when we sampled things the other day from our environment where we just swabbed whatever, all of those things that we swabbed were called fomites. And so in the context of medicine, we have specifically called what's known as nosocomial infections, which specifically are happening in a healthcare setting. And these diseases have specific risks and specific characteristics, but also they're characterized specially because we want to be able to study and understand how to control and mitigate these kinds of infections, because they are occurring within a medical situation where people are already at risk. And so it is a fairly high there's a fairly high danger with these kinds of infections. And so anyway, these infections happen at or acquired at medical facilities. They happen after procedures. They happen like during catheterization. They happen during surgery. And they happen fairly commonly. And there's a risk that because of the environment that you're going to potentially be exposed to even worse pathogens than you would typically in your regular environment, specifically antibiotic resistant bacteria. And so then we have the study of epidemiology, which is studying how these diseases transmit across populations of people. And there are multiple different ways that we do this in the US, where we have surveillance programs and reporting programs which are mandated that as people are diagnosed of these specific diseases, that they are reported specifically to the CDC, and the CDC gathers and aggregates this information to understand when and where are infections happening, what is a concern, and what we need to be looking out for. And so they put this together in this mobility and mortality report. We've got this kind of understanding about this reporting system with COVID in their reporting system. However, they don't just do it with COVID, they do it for all infections. And they also do this with like drug safety as well, and like vaccine safety. But specifically, we're talking about diseases. And so they monitor this and make sure that there are specifically not like something to be on lookout for. They look for, are these cases, is there a pattern emerging in these cases? Could there be potential cause for them? And does some action need to be taken, specifically like for like food recalls or something like that? So depending on where these cases occur, you can classify them in a few different ways, where you have endemic, where they happen in one specific location for a long period of time, where they're characterized within that region, where you say, at those locations, you're going to run into that person. You're going to get one of those infections. That's kind of the vibe. Not necessarily like guaranteed, but that's the idea, right? And so like an example for this would be like, I don't know, I don't have any pathogen examples, but you can kind of think about how across the US, tornadoes are common within this South Great Plains region. And those tornadoes are endemic to that region. You're probably not going to be running into those tornadoes over here, so your risk is less. And so that's kind of the idea of endemic, is maybe infections are happening here and there might be a reason why. Then you have sporadic, where they're happening just kind of all over the place in random intervals, and there doesn't seem to really be any like real causal reason. Now I will say that with with more research or more investigation, there might be a specific cause. Maybe all of these people are eat a specific brand of tofu and that has I don't know, some kind of listeria and contamination or something. And so but you wouldn't see that on just a map, but you'd have to see that more. You would have to do more epidemiological like research into what these cases are in the background of these cases when it's sporadic. Then you have epidemic, which are epidemic is when you have it kind of all over the place within the country, and the cases are drastically increasing in prevalence. And then pandemic is a similar concept, but instead of on a country stage, it's on a global stage. And with that, we conclude chapter 13. I hope that everything was clear, and I will see you in the next one. Bye-bye.